Hi, my name is Bhumika, and this chapter is going to be the part 2 series of the previous lesson that I've done. In the previous lesson, I have concentrated on the structure of the basic components of the nucleic acids. Here, we're going to do the structure of the DNA, and if you want to check out the previous lesson, then you may do that as well. And I would actually advise you to go through the first lesson because that would make you understand this chapter well. Okay, so let's jump right into it. Right, we begin with the structure of DNA. I have already explained the basic components. That means the phosphate group. I've explained the ribose sugar, and then I've explained the nitrogenous bases as well. So you may go and check that out. Right. Here, what we've got is the Watson and Crick model of the DNA. DNA stands for deoxyribonucleic acid. And why is it called deoxyribonucleic acid? We'll understand in a minute. All right. So we have this chain right here. As you can see, we've got some phosphate atoms connected to a pentose sugar. Now, this pentose sugar and the phosphate group, they form the sugar phosphate backbone, right? So the sugar phosphate backbone is present parallel to each other, and in the center, as you can see, these in green, they are the nitrogenous bases. I have already explained the nitrogenous bases in lesson 1, so for your reference, you can check it. Right. So we can see that if we magnify this version of this pentose sugar right here, this is a deoxyribose sugar. Okay, and I have numbered the carbons starting from this point. So this is 1 prime, this is 2 prime, 3 prime, 4 prime, and 5 prime. Now, as you can see, a phosphate group is attached to the 5 prime carbon and the 3 prime carbon at one end, and on the other end, it's the 5 prime carbon. If we go on and number all the carbons of these series in such a way, we'll see that on one side the 5' end is free, and on the other side the 3' end is free. Therefore, the polarity of this strand is 5' to 3'. On the opposite side, on one side the 3' end is free, as well as on the other side the 5' end is free. Therefore, the polarity of this strand is 3' to 5'. And thus we say that both the strands are anti-parallel to each other. If one strand is 5' to 3', the other strand will be 3' to 5'. Okay, now let's talk about the nitrogenous bases which are present in the center. On the 1' carbon, we've got thymine, which will be doubly hydrogen bonded to adenine. Then we've got another thymine, we've got another cytosine, which is triply bonded to guanine, and then guanine to cytosine. Now, this is a very, very small segment of the DNA. This is, you can call it an oligonucleotide. So basically, an oligonucleotide is when a few number of nucleotides are joined together to give an oligonucleotide chain. But if I join some of the hundreds or 200 or 300 or more than that nucleotides together, they give rise to a chain which is known as the polynucleotide chain. This polynucleotide chain is the core structure of the DNA. All right, moving on, we've got the phosphate group here, and the pentose sugar here, and then we've got the bases. Okay, this picture is the courtesy of a BBE representation, and I got it from Google. This is not my picture, and I'm claiming no rights for it. Okay, so now I told you that these bases and these chains are composed of 100 and more than that, perhaps, nucleotides. So when so many nucleotides are bound together in a chain, it produces tension. When it produces tension or strain in the chain, it coils itself in such a way. Now, this DNA, this thing right here, is the double helix model, which was very fondly explained by Watson and Crick. Okay, moving on. We've got the salient features of DNA. These are ultra important, and they many a times asked in the examination. All you've got to do is you've got to explain the structure of DNA, and that too in very precise points. So it contains two polynucleotide chain wound around each other. Okay, I would like to make sure that this diagram is not for your reference. I've just drawn it to accentuate the look of my spread. Do not refer to this diagram. If you had to, you will refer to this diagram or the diagram which is drawn in this picture, and nevertheless this one. Okay, so we've got the first point, as it contains two polynucleotide strands wound around each other, as we saw in the previous picture, two polynucleotide strands which are wound around each other. Now the backbone of each consists of alternating deoxyribose and phosphate groups. Pretty clear from the structure that we just did. Now the phosphate group is bonded to the 5' carbon atom of the first deoxyribose, and it's covalently bonded to the 3' carbon atom of the next. We did it here, 5' at this point and 3' at this point. Okay, the two strands are antiparallel, that is, one runs from 5' to 3' and the other one runs from 3' to 5'. Now, the purines and the pyrimidines are attached to each deoxyribose, and they project in towards the axis of the helix. Axis means the central portion of the helix. All the nitrogenous bases, the adenine, guanine, thymine, and cytosine and uracil in case of RNA, they are present in the central portion, in the axis of the helix. Now each base, it forms hydrogen bond, with the one directly opposite to it, forming base pairs. Alright, so adenine and thymine will form double bonds, giving rise to a adenine-thymine base pair. Alright, now the diameter of the helix is 20 angstrom. The helix can be virtually any length when fully stretched. It is at max, it can also be almost 6 feet. That's the maximum amount of DNA that can be extracted from a cell, and a cell is so small, it's microscopic, so you can imagine the complexity of it. The path taken by the two backbones form a major wider groove and a minor narrower groove. As you can see, this is the minor groove, which is narrow, and then we've got the major groove, which is far and wide. So that's about the DNA. I would definitely recommend you checking out the first tutorial, because it has the details of the bonding which is there, and the purines and the pyrimidines, about the bonding that they occur with each other. Yeah, thank you so much for watching, and I hope you check out the other tutorials as well.